RNA viruses are a little bit different. Remember, RNA doesn't have to go into the nucleus in order to be transcribed. It's already there. So RNA viruses tend to be found only in the cytoplasm of the host cell. They often have to encode their own RNA polymerases because our cells don't have a mechanism to read RNA and then copy it into RNA. We don't have that type of replication. So RNA viruses bring a lot of their own machinery with them. One of the other things about RNA is that it is relatively unstable. So it's a little bit more transient as a molecule. Most RNA viruses will replicate inside the cytoplasm. And again, like I mentioned, our cells can't replicate RNA. So RNA viruses have to use what's called an RNA dependent, so it reads RNA polymerase to make RNA. That's very, very different from our host cells. And when we have things that are very, very different from our host cells, sometimes that allows us to have a target for treatment. The genome structure determines exactly how transcription and replication occur. Single-stranded versus double-stranded, positive sense strand versus negative sense strand. The genome structure and polarity determine how the viral messenger RNA is generated and proteins are processed. Is it plus or minus strand? RNA viruses are prone to mutation. The polymerases that replicate the RNA genome are not as good. All RNA viruses, except for a positive sense RNA genome, have to bring a polymerase with them. So they're packaged with a polymerase. The reason that positive sense strand RNA viruses don't have to bring a polymerase with them is because their genome basically serves as a transcript. So as long as their genome encodes a polymerase, our cells will make the polymerase for them. Then they would use that polymerase to replicate their genome. And as far as we know, all negative sense strand RNA viruses are enveloped. Viral genetics can be a little bit complicated. You wouldn't think so, but they can be. So here are a few different things that we can see with viral genetics. And I think most importantly, we're going to talk about just 1 and 2. But again, the idea is that viruses can, if they're very closely related, can mix and match. So herpes simplex 1 and 2 can sometimes mix and match. Some viruses have segmented genomes. So instead of having a single piece of DNA or RNA that represents their genome, they have multiple pieces, almost like chromosomes. For example, influenza has 8 different pieces of genetic material. And what we see is that we can get reassortment. So if this is flu strain 1 and this is flu strain 2, and they both infect the same host, you get a little mixy-matchy, and now you have 2 completely different flu strains that come out. We also can have certain times where a virus that has a mutation could recombine with a wild type and become infectious again. And we'll see that with the polio vaccine. What type of disease we see depends on where the virus will infect, how the virus gets in, how pathogenic that particular strain is. And the severity of the disease, again, can depend on a lot of different factors. And as we talk about each infection, we'll talk more about these. With progression of disease, you have acquisition, initiation of infection, activation of innate immune system. Then you'll have an incubation period where the virus could potentially spread to a secondary site. You'll have replication in the target tissue, which will cause the characteristic signs and symptoms. So, for those of you who are going into the medical profession, signs and symptoms are different. Signs are things that somebody else can see. So a sign might be diarrhea, a sign might be a rash, a sign might be hemorrhaging. whereas a symptom is something that you as the patient feel and have to describe to your physician. So let's say you have a rash, that's the sign. The symptom could be itching or pain because your doctor can't see that. If you have diarrhea as the sign, a symptom could be abdominal cramping or nausea. If you have hemorrhage, again, the doctor can see the hemorrhage, but you might feel pain or lethargy. So those are the differences between signs and symptoms. Okay, then there's host responses that can both limit the infection but also contribute to the disease manifestation. And then as the virus is being produced and released, that could make that person contagious. And with a viral infection, you could have resolution where the infection goes away, or you could have persistent infection or chronic disease or even host death. Most viruses that we have in humans get into the body through inhalation. Of course, there's also ingestion, breaks in the skin, and that would include viruses that are transmitted by insects. They'll generally break the skin. If virus is being transported in the blood, that's viremia. But viruses, remember, can't replicate in the blood unless they are in a blood cell that allows replication. And there are some viruses that can replicate in our white blood cells. Secondary viremia is if the virus has already replicated in various tissues and cells and then re-enters the bloodstream. There are 4 major outcomes of infection. You can have a failed infection. They call it abortive infection. You can have cell death. You can have replication of the virus without cell death. And you can have the presence of the virus without production, but with the potential for reactivation. And that one is a lot of fun, and we'll talk about that. How a virus interacts with the target tissue depends on a lot of different factors. And again, as we talk about the different viruses, we will talk about all of those things. Now, one of the things for those of you who may be interested in like CLS that you have to be aware of is what are the cytopathological effects of the virus on host cells. So basically, if you look at an infected host cell under the microscope, can you diagnose the infection that way? And there are certain viruses that will do very specific things to host cells that you can see under a microscope. And depending on the virus, depending on a lot of different factors, what it does to the host cell, again, is different for each virus. Our immune response, we're going to have an innate immune response at first, antigen, non-specific, antiviral responses. As we talked about when we talked about immunology, one of the major ones is interferon and other cytokines. Natural killer cells and macrophages can also be important here. As the infection progresses, eventually you will develop the antigen-specific or adaptive immune response, which will include T cells and antibodies. And just like with everything, we have evolved mechanisms to fight viruses. Viruses have evolved mechanisms to hide from us. So there are a lot of immune evasion strategies we can talk about. A lot of the symptoms that a patient has from a viral infection can result from the immune system rather than viral action. So flu-like symptoms, which include fever, headache, tiredness, are often a result of interferon production. But we can have T cell responses that lead to cell killing and inflammation. Antibody responses can also lead to cell death, and that can cause inflammation, or what are called immune complexes, which can cause swelling. And there are other inflammatory responses involved. One thing that's really interesting about viral infections is a lot of them are associated with causing cancer. We talked about one really, bacterial infection that can cause cancer. We talked about Helicobacter pylori, but there are a lot more viruses that can be associated with causing cancer, and we call those oncogenic viruses. This is a set of the immune factors that can lead to disease symptoms or signs. So flu-like symptoms, inflammation, hemorrhagic disease, cytokine storms, immune suppression, and some of the viruses that can cause those signs and symptoms. And again, as we talk about the specific viruses, we'll talk about how that all occurs. As I've mentioned before, the disease severity will result from a combination of different factors. No one person is going to experience the exact same disease that you experienced. And I think right now, living through a pandemic like COVID-19, we've all seen that. So COVID went through my house, and every single person in this house had the infection slightly different. Longer course of infection, shorter course of infection, higher fever, lower fever, no fever. It really all depends. And that was really interesting because I have twins. So you would think that, overall they're kind of similar, but they're fraternal. So their genetics are different. So it's entirely possible that that's why both children had different effects. So the mechanism of exposure and site of infection, the immune status, age, and general health of the person, the viral dose, the genetics of the virus, and the genetics of the host. Here's a little bit more detail about what viral diseases can look like. Most commonly, you'll see an acute infection where you have your disease episode, you get better. More rarely, you'll have an acute infection followed by late complications, and this would be like decades later. This is seen with measles. They'll have the initial measles episode, and then decades later can develop something called subacute sclerosing panencephalitis, which is very, very dangerous. Latent recurrent infections are also fairly common. You'll have an initial disease episode followed by disease episodes later, and that is what we see with varicella zoster, chickenpox, and shingles, or the herpes simplex viruses. We can have chronic infections with things like hepatitis B and C, chronic infections with a late disease. That would be something like HIV. A patient who has HIV will develop initial flu-like symptoms, will feel much better, and then eventually will develop AIDS. And then finally, we have very slow infection. The one that we're not going to talk about JCV, but we are going to talk about prions. So Creutzfeldt-Jakob, this could be decades after exposure before the disease finally shows up. Viruses can be transmitted just like bacteria, number of different mechanisms. A little bit different from bacteria is genetic transmission. So retroviruses have genetic transmission. And then the factors that promote transmission include the stability of the virus, the replication and secretion of virus, asymptomatic transmission, and how the immune response works. Our risk factors are basically the same risk factors for any infection: age, health, immune status, lifestyle. Are your kids in a daycare center? Apparently those are big germ factories. And of course, your critical community size. How many people in the community are susceptible to the infection? That can really have a big effect on how a virus or any disease spreads through a population. And depending on the mechanism of transmission, geography and season can play a pretty big role. Anything transmitted by arthropods, you're probably going to see it at higher levels in the summer or in a wet season, because that gives the best available environment for the vector. During school, we tend to see higher levels of infection because of close proximity and crowding. Home heating seasons, basically during the winter, you tend to just be more susceptible to disease. Again, probably because of that crowding, the increase in temperature in the home. And there are lots of mechanisms of control. Of course, the most important, vaccination, but also quarantine, education, and if it's an insect transmitted disease, elimination of the vector.